Okay, so really briefly, I mentioned in the last video these things called antibodies, and what are antibodies, and why do I need antibodies, and what do they do? So you need to know from the dot points, and you can read this dot point on the top of page 13. It says you need to know the actions of B lymphocytes and their antibodies, including antibody structure. So I must teach you the structure of antibodies to get through this. So let's have a look. We're down now on page 15, and you get to see this guy here. So this is your classic antibody. You do not have to draw them, though, really complicated. You can just draw them like this. So I draw like that. Short chains here. So there are 4 kind of chains that make up the Y. Then we've got a shape here that's the same. And if you want to, you can put in this guy down here. But that is sufficient when drawing an antibody. And even though I've got it all kind of like scribbly, it's not doing palm rejection, that's good. So even though it's all kind of scribbly, that's what we're going for. Actually, I'm going to do this on a big board just one more time, just to show us. So you go down like this, and they have on past exams asked you to draw an antibody. So they are 4 chains. These guys are, that's not a great side for me. Oh goodness, I am not having fun with this today. Short. So these guys are proteins. They are made up of 4 chains, so they are actually a quaternary protein. Quaternary. Why can I not spell today? Why can I not spell today? I cannot spell today. Quaternary. Oh my God. You get what I mean. I'm just going to do this because it's going to save me, and you can just laugh. You can just be sitting there and laughing at me. There we go, quaternary proteins. They have 4 chains. They have 2 heavy chains and 2 light chains. Now the heavy chains are obviously my long chains. They're these guys here. And the light chains are these guys here. Now the other thing that we have on an antibody is we have 2 of these bits up here. And they are called your antigen binding sites. Okay, so there you've got your antigen binding sites. You've got 2 of those. And then the last thing that we often talk about is that this bit up here is what we call the variable region. But I've never seen you being asked about this, by the way. And this pink's nice. This bit down here is known as the constant region. So I'm just going to take a little screenshot of that so I can send it to you later. So that is how you do your antibodies. So here are the antibodies. We've got more of them up here. You can see there's different images to show you the same information. So you've got 4 chains. They don't have to be chunky. They can just be lines. This one's got 4 chains. They always form this Y structure. They have this antigen binding site that is complementary to the shape of the antigen. And there are 2 of them. Now, the other thing you might find is some of them talk about these disulfide bridges. You don't need them. Okay, you don't need to mention them, so it's okay. Here is another picture. You can see this is nice and simple. So in your notes on page 15, these guys are the things that are produced by your B plasma cells. They are made of protein. The biomacromolecule that these guys are made of, that is their macromolecule, which is a question that got you on one of my EP tests. So the macromolecule they are made of is a protein. Each has a specific antigen binding site, and that is complementary in shape to the antigen. So these are all very, very, the other thing that I haven't written on here is they are super specific. Okay, super, super specific. They're not always like, usually it's one type of antibody per antigen, but sometimes you can get antibodies that fit to multiple antigens, but it's very similar in shape, those antigens. So this is what we're talking about here. This is if you get two similar shaped antigens, the antibody will work against it. Now, this is technically off the study design. However, last year, they asked a question that kind of needed for you to understand this. So I don't want to leave you unprepared. They gave you just enough information for you to kind of infer it last year, but it wasn't good enough. So what I'm saying is that an antibody usually binds to one antigen only, except if that antigen, can you see how antigen 1 here and antigen 2, these guys have very similar shapes. If they have very similar shapes, then what you're going to end up with is the antibody might work on both. The other thing you've got these things called epitopes. Now, this is definitely off the study design, but I just want to show you what we're talking about here, is that one antigen can actually have multiple shapes to it, and so different antibodies can bind to it. But well off the study design. I just thought if I introduced the word, if they decided to tell you about them in the exam, then that would be okay. This, again, is also, the names don't have to be learned, so you don't need to know IgG, IgD, and IgE at this stage. We do talk about some of these. We talk about IgG ones when we talk about the allergic response in the next unit. But immunoglobulins, these, again, there's a reason they're not in your notes, and it's because you don't have to learn them. Okay, but there's one that's called the IgG one. It's a specific antibody, and it helps to speed up destruction and of other, helps to identify pathogens and speed up their destruction. IgD, this is one that seems to be bound on the surface of a B cell. We've got IgE. You can see all these different ones that we talk about. So IgE, is the one that we're going to be talking about when we come into allergies. So this is the only one, I think, that we really need to kind of think about. And even then, you don't need to know. You can just call antibodies. Then you've got IgA. You've got these ones. These are the ones that are in your tears and saliva, and all those kind of inches. And then you've got IgM, which is this big kind of ninja star-shaped one, which can bind to lots and lots of bacteria. So they are all in your book. I'll let you go back and have a read of that if you want to, but you don't need to. So how do they work? And this is where we go down our 4 things. It's what I like to call NAPA. Okay, so NAPA. They can neutralize. So what they can do is the antigen binding site can bind to the toxins, and they can also bind to the pathogens and stop them from entering cells. They can then do this thing called agglutination, which means binding together. It means sticking together. So agglutination is best explained in a picture, and I'll show you that on the next one. Then we've got precipitation, which is where you bring them out. If you're a chemist, you'll know what precipitation is. So you've gone from the antigens and the pathogens floating around in your body fluids. If you grab them with antibodies and stop them from moving, then they might remove themselves from the liquid and become a solid, which makes them much easier for your body to deal with. And the last thing they can do is they can activate complement proteins. But if you can just remember those first 3 as well, that's really good. Why have I got CNAP? Oh, CNAP for complementing proteins. So you can call it, I like NAPA, but I think CNAP is better because you don't know what the difference between the A's is. So the C is for complement proteins, the N is for neutralization, the A is for agglutination, and the P is for precipitation. So this is what I'm talking about with the neutralization. So the neutralization masks the agglutination. So here is the antibody. Can you see how this antibody here can bind to all these different pathogens? And they kind of stick them together. When they stick them together, they're also going to make them a big clump. Now, when that happens, what you get to do is you get to be eaten much more easily. That's great. If you do manage to activate the complement proteins, though, what they can do is they can end up bringing more of these phagocytes together, more phagocytosis. And the other thing the complement proteins can do, because remember there's like 30 of them, is they can cause the cell to be burst on its own. Now, the last little bit that I have here is about this thing called clonal selection theory. Now, it is off the study design, but they have asked questions about it, so we're going to go through it so we understand. All is it's saying is that only, you can have a read of this one in your book as well, only immune cells that are activated by the T helper cells will divide. None of the others will divide. Okay, and what happens is they divide rapidly, and that's so you get a rapid response. And this happens in the lymphatic system. Lymphatic system. So this will happen in your lymph nodes. Whenever you get exposed to something, it'll find it and it'll go, oh, this is the cell that I want to expand. And then all of a sudden, bang, and clonal selection and expansion occurs. So here's the question from a couple of years ago on the exam, this study design, and this is what they asked. So they said the lymphatic system includes lymph nodes, spleen, and tonsils. In particular, these organs, clotting factors are inactivated to help seal a wound, but clotting factors are inactivated is definitely going to be wrong, because if you want them to be activated to seal a wound. Clonal selection and proliferation of B cells occurs. That sounds pretty good. So cloning, the other word for it is proliferation, and I really hate saying it, so I'm saying it slowly. Proliferation. Clonal selection is just many, many copies of something happening really, really fast. So that is correct, but let's just check. Non-self antigens are identified by red blood cells. No, because they would be identified by white blood cells. And does the lymphatic system respond to allergens? No. Now we're going to talk about that. Yes, it does, but no, not the lymphatic system isn't responsible for your allergies. So what it is with this one is that you get clonal selection and that proliferation of B cells. And that is the end of my little talk about antibodies.